In Chapter 6, we are going to cover the properties of metabolism. Virtually every living organism on planet Earth consumes energy in some form. Think about plants, for instance. Plants undergo a process known as photosynthesis, and without light from the sun, their predominant energy source, plants are never going to be able to produce energy. Plants will never be able to grow. Plants will never be able to reproduce. That would be very detrimental for us as humans and our overall environment, because we know how essential plants are, not only for their role in oxygen production, but also their role as a food source. So a lot of consumers, a lot of animals eat different types of plants. There's an intricate interaction between plants and fungi, and plants and bacteria, and plants and many other organisms. So a lot of the organisms depend on the production of energy and the production of the life cycles, from the life cycles of plants. So let's talk about bioenergetics. This is the study of energy flow through a living system. So when we talk about bioenergetics, we're going to talk about all the cellular processes that happen within the cells. So our cells are constantly building up and breaking down complex molecules through these stepwise chemical reactions. Some of these reactions are spontaneous and they release energy, where others require energy to proceed. So as we get through this chapter, you're going to be able to understand and differentiate between some of the essential chemical reactions that occur inside cells, including those that use and release energy, and therefore you will gain a better understanding about cellular metabolism. Metabolism refers to all chemical reactions of a cell or organism, and a metabolic pathway is a series of biochemical reactions that converts one or more substances into a final product. So let's take a look at this picture down here. On this side of the picture, this is an oak tree, and you can see some acorns on this oak tree. The oak tree and the acorns use energy from sunlight to make sugar and other organic molecules. And both plants and animals use cellular respiration to drive energy from the organic molecules that plants originally produced. So this squirrel depends on the cellular energy that was originally produced by the acorns over here. And as you know, squirrels depend on acorns as a predominant food source, especially in the winter months. So they're going to be hoarding acorns wherever they choose, and they will be able to crack the acorns open and get the energy out that the plant once harnessed. We're going to talk all about photosynthesis and how the chemical processes and the chemical steps of photosynthesis occurs, and we're going to learn all about cellular respiration as we proceed through this lecture and future lectures. Now I want to introduce you to two of the major metabolic pathways. So we have metabolism, we've already talked about being all of the chemical reactions that occur inside a cell. Now we're going to specify of whether we are taking small molecules and building them into larger molecules, or taking large molecules and breaking them down into smaller ones. Okay, it's very important in this slide to pay attention to where energy is required and where energy is released. So let's first talk about anabolic reactions. So you may have heard the term previously, anabolic steroids. So anabolic steroids such as testosterone or Winstrol or Duranabol. These are common substances that are abused by bodybuilders and in some cases athletes. So anabolic steroids, what they do is they help to take small molecules and assemble them into larger ones. So they will help to build muscle. So again, small molecules are assembled into larger ones. And for this process to happen, energy is required. On the opposite end of the spectrum, we have catabolic reactions. So this is when these large molecules are already assembled and they are broken down into smaller ones. During this process of breakdown, energy is going to be released. So again, please be sure that you're able to differentiate between anabolic reactions and catabolic reactions as far as what energy is used for. Anabolic energy is required to build the larger molecule, while in catabolic, energy is released as the larger molecule turns into the smaller molecules. So this picture here shows the tree of evolution of various branches of life. So we're going to start here at the bottom with the prokaryote ancestor, and the vertical dimension is time. So as time progresses, the tree branches and different organisms have formed. What's interesting about all these organisms is that all types of life share some of the same metabolic pathways. So us as animals up here, we share some of the same metabolic pathways with protists and with plants and with fungi and with archaebacteria, eubacteria, prokaryotes. We have some things in common, so this gives some credibility to the theory of evolution. But over time, you all know that we are more complex cellular beings than prokaryotes. We are considered to be eukaryotes. So our cells undergo much more complex processes, and we have developed special proteins and other special anabolic and catabolic pathways to help us live and sustain the necessary homeostasis and the homeostatic balance that our body needs in order to be functional and survive. So now a little discussion question for you all. Let's talk about photosynthesis, and let's introduce the key elements and compounds in the photosynthetic reaction. So is photosynthesis an anabolic or a catabolic pathway? So again, photosynthesis is driven by solar energy. Without the sun, photosynthesis does not occur. You can always use artificial lights to artificially induce photosynthesis as well. But for generally speaking, photosynthesis is driven by the sun and the sun's energy. Okay, so let's talk about some of the key compounds in the photosynthetic reaction. So again, on the left side of the reaction, we are going to have the reactants, I'm going to term as R. And on the right side of the reaction, we are going to have the products that I'm going to term as P. So we have carbon dioxide, and I'm going to talk about what these numbers in front of carbon dioxide and water and oxygen mean in a little bit, and why we have to stoichiometrically balance this equation. So we have carbon dioxide plus water, and that is going to yield us sugar or glucose and oxygen. So I'm going to give you a second to think about this. Is this an anabolic reaction or a catabolic reaction? So for those of you that said anabolic reaction, you are correct. We are taking two simple compounds in the reactant side, and we're making a very complex molecule in terms of glucose, and oxygen is pretty much just our byproduct. Okay, so I'm going to erase some things now, and we're going to talk about what all these numbers mean. So I just want to erase things so I don't have a bunch of scribbles all over the screen. So let's start with carbon dioxide. Well, let's talk about carbon specifically first. So we have to be stoichiometrically balanced, and what that means is we have to have the same number of molecules of each element on our reactant side as our product side. So these both have to match. So let's start with carbon. Okay, so carbon out here, there's no subscript under carbon here, so that means there's just one carbon molecule. But there is a 6 out here in front of carbon. So we do 6 times 1, and that's going to give us 6 carbons on this side. Now let's go over to our product side. And if we look in the glucose compound, well, we have 6 carbons already. See this, the subscript 6 here, 6 carbon molecules here. So we have 6 carbons on this side. So carbon's nice and balanced. That's why we have to have that 6 in front of carbon dioxide. Now let's talk about oxygen. So oxygen on the left side of the equation has a subscript of 2, meaning 2 molecules. So what we have to do is we have to take that 6 times 2, and that's going to give us 12 oxygen molecules. for carbon dioxide, 12O. But we also have this oxygen as well. So we have to do 6 times 1, and that's going to give us 6 more oxygen molecules, which is going to give us a total of 18 oxygen molecules on the reactant side of the equation. So now let's look on this side. Well, in glucose alone, we have 6 oxygens. So glucose alone, we have 6. And then again with oxygen, we do 6 times 2, and that's going to give us 12 oxygen. And 6 plus 12 is going to give us 18 oxygen. So our oxygen is balanced on both sides. So now let's look at our hydrogen molecules, and let's see if hydrogen is balanced on both sides of the reaction as well. So give me a second, I'm just going to delete everything for clarity purposes. Thank you for your patience. All right, so now let's talk about hydrogen. We've already talked about oxygen. So here we have 6 times 2, and that's going to give us 12 hydrogen on the reactant side. And already nicely in the glucose compound are 12 hydrogen molecules. So we have 12 on each side. They're balanced. So that's why we have to balance the equation so that we have equal and opposite amounts of molecules on each side, because again, energy is neither created nor destroyed. And this is going to be the thinking question down here. What is the opposite process of photosynthesis? We're going to talk about this in the upcoming lecture. So if you guys want to get ahead, think about what would be the opposite process. How could we take glucose and oxygen, and what would we do to those? Do we build them up further, or do we break them down? Now that we've introduced energy, let's define it and talk about some of the subclassifications of it. So energy, by definition, is the ability to do work, and it can be classified as either kinetic or potential energy. So objects in motion have kinetic energy. So in this picture here, you can see the water running down the waterfall. This water is said to have kinetic energy. But let's look at the picture on the left side here, water that's behind this dam here. Well, that water is not necessarily moving or flowing in a sense. It still has energy in the form of potential energy. Let's say this dam was removed or there is destruction to the dam, well then the water could flow and become kinetic. Now I want to give you all some examples of potential and kinetic energy in cells. So remember on a previous lecture when we talked about the phospholipid bilayer of the plasma membrane, there are many molecules and substances and ions that are constantly going in and out of this membrane. So the energy of the electrical chemical gradient across the plasma membrane is an example of kinetic energy. There's constantly all sorts of things moving in and out of the cell. Chemical energy, energy stored in chemical bonds, this is going to be a major type of potential energy. This is one of the biggest examples of potential energy in cellular reactions. So all of the energy within these bonds here, so you can see this oxygen bond here, once this oxygen bond is broken, then energy can be released and the energy then becomes kinetic. So energy stored in chemical bonds, that's the potential energy. When those bonds are broken and the energy is released, it becomes kinetic. And in gasoline, also known chemically as C8H18 or octane, gasoline or octane has its energy stored in chemical bonds as well. So all the bonds between the carbon molecules and all the bonds linking the hydrogen to the carbon molecules. This is the energy here is in the form of potential energy, and it's transformed into kinetic energy once the engine in the car system uses that gasoline to propel the car forward. And once it transforms that energy, it is then turned into kinetic energy, and again, it allows the car to accelerate. Now that you have all learned that chemical reactions release energy when that potential energy that's stored in the bonds breaks or dissociates, now let's think of another very important question. Let's see if we can answer this question. How do we quantify and express the chemical reactions with the associated energy? So this energy that's being released from these broken chemical bonds, how do we quantify it? How do we know how much is there? How do we determine what type of energy that is? How can we compare that energy that releases from one reaction to that of another reaction? Well, the way we do that is we use a measurement of free energy. Scientists call this free energy Gibbs free energy. And Gibbs free energy is abbreviated by the capital letter G. And this is the amount of energy available to do work, or this is the amount of usable energy. So every chemical reaction involves a change in free energy. And whenever you see this delta symbol in front of a letter, that means change. So delta G means a change in the amount of usable energy. So this equation down here is really important for you all to know. This is going to be the formula that you use to calculate the change in free energy for any system. And this is used to calculate the energy in many different types of chemical reactions. So to calculate the change in Gibbs free energy, you subtract the amount of energy lost to entropy, which is denoted by this change in S, or delta S, from the system's total energy change, which is denoted as delta H, or the change in the total energy of the system. And then T is going to be your temperature in the unit of kelvins. So again, this is going to be the formula for calculating the change in G, or the change in Gibbs free energy. And we're going to talk about what this means and why this is relatively important on the upcoming slides. So now let's talk about what these G values mean and why they're important to understand. So if energy is released in a chemical reaction, then the change in G is going to be negative. It's going to be less than 0. So this is because the reactions that release energy have a delta G value less than 0. A negative delta G also means that the reaction's products have less free energy than the reactants. So because they gave off some type of free energy during that reaction. If a chemical reaction requires energy to proceed, then the delta G value has to be positive, or it has to be greater than 0. So in this case, the products are going to have more free energy than the reactants. Again, remember back to the previous slide. If delta G is less than 0, or if delta G is negative, then the reactants are going to have more free energy than the products. Okay? So please don't confuse these two areas. If delta G is greater than 0, or it's positive, then the products are going to have more free energy than the substrates. And these reactions are called endergonic. These reactions are non-spontaneous, and these reactions will not take place on their own without adding free energy. So energy, I'm going to write it out here, is a requirement. Energy is an absolute must here. If we don't have energy, these reactions are not going to proceed. They are not spontaneous reactions like those that we see when the delta G value is negative. So now let's move into another discussion question, and I hope you all haven't forgot what we learned about dehydration, synthesis, and hydrolysis reactions. So let's take a minute, and we're going to term this first reaction number 1, and we'll term this reaction number 2. Which one is an example of dehydration synthesis? If you said reaction number 1, you are correct. This is an example of dehydration synthesis, and what we're making out here is the molecule glucose. And by default, number 2 is hydrolysis. So we're using hydrogen and oxygen in the form of water to hydrolyze this bond here, or break this bond and make 2 separate molecules. So another question, is reaction 1 or reaction 2 the anabolic reaction? If you said reaction 1 is the anabolic reaction, you are correct. You're taking 2 simpler molecules and you're making 1 more complex molecule here. And by default, the hydrolysis reaction is going to be a catabolic reaction. So we're taking this complex molecule and we're simplifying it into 2 simpler molecules. So which one is exergonic? Which one is going to require energy to do? Which one is going to have a negative G, delta G, and which one is going to have a positive delta G? So I'm not going to give you that answer. I want you to research that, and we're going to talk about that in the discussion questions this week. So there's another important concept that we must consider regarding endergonic and exergonic reactions. Even exergonic reactions require a very small amount of energy before they can actually proceed with their energy releasing steps. These reactions have a net release of energy, but still require some initial energy. So that's where the whole subclassification of potential and kinetic energy comes to application here. So scientists call this small amount of energy input necessary for all chemical reactions to occur activation energy. So activation energy is the hump in the diagram. So this hump here and this hump here. So whether the activation energy is of the catalyzed reaction or the uncatalyzed reaction, that is going to help determine the overall energetic output and the energetic state of the reaction. So again, this here is going to be an exergonic reaction, what we're looking at here. This reaction, exergonic, delta G is going to be less than 0, delta G is going to be negative. This reaction is spontaneous. But as I mentioned, spontaneous reactions can occur. They do not need energy to occur, but they do need that initial activation energy to get them going. Once they get going, they do not need energy to proceed any further. So what the activation energy actually does is it causes the reactants, so remember, the reactants are on the right side of the equation and the products are on the left side of the equation. So it causes the reactants to become contorted and unstable. And what that does is it breaks those bonds. So all that potential energy that's stored up in those bonds then turns into kinetic energy. This unstable state before the bonds are actually broken is known as the transition state. And the transition state can occur very, very quickly. So heat energy is going to be the main source for activation energy in a cell. So think about this. What happens to molecules when you heat them? They speed up, they start to move faster, they start to collide with one another. And the heat is essential for helping these reactants reach their transition state. It helps them reach this unstable state so that no additional energy is needed after that point for the reaction to occur. So again, we need that initial activation energy. So even in exergonic reactions, so we need, I'm going to change colors here. So we need this initial green energy, right? So this is the catalyzed reaction. This is the reaction that is mediated by this whole aspect of activation energy. But once we reach a threshold, the reaction can become spontaneous because we don't need any energy here. This is the activation of the uncatalyzed reaction. So again, this is why I said earlier that the exergonic reactions do not need energy to proceed. They do need an initial activation energy, which is going to be in the form of the catalyzed reaction, But once the catalyzed reaction causes a threshold to be released, which is indicated by these dotted lines right here, then the uncatalyzed or the spontaneous part of the reaction occurs and the reaction can proceed without any further energy. And again, bringing back the gasoline or the octane example, this again is an exergonic reaction. So gasoline, in order to break these bonds, to break all the bonds between the carbons and the hydrogens and the carbons and the carbons, some initial event has to happen. Well, that spark is required. It's required to turn the ignition and press the gas pedal in order to provide sufficient heat to exceed the activation energy. So again, once that activation energy is needed, the rest of the reaction is going to become spontaneous. Energy is going to be released, and the energy that is released provides enough heat to drive any type of necessary additional reaction. Thermodynamics refers to the study of energy and energy transfer involving physical matter. The matter and its environment relevant to a particular case of energy transfer are classified as a system, and everything outside that system is the surroundings. So for example, think about heating a pot of water on your stove. The system includes the stove, the pot, and the water. And energy transfers within the system. Energy transfers between the stove, the pot, and the water. There are two types of systems. We have open systems and we have closed systems. An open system is one in which energy can transfer between the system and its surroundings. Makes sense, it's open. It can leave its initial location, it can go into its surroundings or into the environment. And the example I gave you all about the stovetop is considered an open system because it can lose heat into the air. A closed system is one that cannot transfer energy into its surroundings. And us as humans, we are considered to be open systems. Energy is constantly being exchanged between us and our environment. We consume energy molecules and we release energy into the environment by breathing and doing other examples and types of work. Like all things in the physical world, energy is subject to the laws of physics. And here we're going to talk about how energy is governed by the first and the second laws of thermodynamics. So let's begin by talking about the first law of thermodynamics. And this law deals with the total amount of energy in the universe. It states that this total amount of energy is constant. So it's always been here and it always will be here. And it's always the exact same amount of energy at all times. Energy is neither created nor destroyed. And it exists in many different forms. So according to the first law of thermodynamics, energy may transfer from place to place or transform into different forms. And again, it cannot be created nor destroyed. The transfers and the transformations of energy take place all around us all the time. Light bulbs transform electrical energy into light energy. Gas stoves transform chemical energy from natural gas into heat energy. Plants perform one of the most biologically useful energy transformations on Earth. They do photosynthesis. So again, energy can never be created nor destroyed. Energy is always preserved. And if you think about Buddhist philosophy here for a little bit about reincarnation, reincarnation may not be meaning that, okay, I'm a human now, I'm going to be reincarnated into a dog or a cat or whatever you have you. But reincarnation may be thought of as my energy that makes me up is going to be transferred into a different form in the next life or once I pass. So something interesting to think about from a philosophical standpoint. Now let's move into the second law of thermodynamics. So down here, second law of thermodynamics. This law has to deal with the fact that a living cell's primary tasks are to obtain and transform and use energy, and to help us understand why these tasks are actually harder than they appear. It should be relatively easy to use energy, but scientifically speaking, this is a relatively complex task. So no type of energy transformation or energy transfer is 100% efficient. There's always going to be some type of lost energy in a form that's unusable. In most cases, this form is heat energy. So thermodynamically, scientists define heat energy as energy that transfers from one system to another that is not doing work. So let's take the example of an airplane flying through the air. Well, as the airplane flies through the air, it's going to lose some of its energy as heat energy due to friction with the surrounding air. This friction actually heats the air by temporarily increasing air molecule speed. And some energy is lost as heat energy during cellular metabolic reactions that occur in our body. This is good for warm-blooded creatures like us because heat energy helps to maintain our body temperature. So again, no energy transfer is 100% efficient because some energy is lost in an unstable form. An important concept in physical systems is that of order and disorder. So when I refer to the term disorder, I mean randomness. So the more energy that a system loses to its surroundings, the less ordered and more random the system. So scientists refer to the measure of randomness or disorder within a system as entropy. High entropy means high disorder and low energy. So let's try to understand entropy a little bit better. Think about a college student's bedroom. If no energy or work were put into it, the room would quickly become messy. It would exist in a very disordered state, one of high entropy. Energy must be put into the system in the form of the student doing work and putting everything away after use each and every time in order to bring the room back to a state of cleanliness and order. This is the state of low entropy. Now let's go back to talking about endergonic reactions, where the energy of the reactants has to be greater than that of the products, yielding us a delta G value that is positive or greater than 0. So how do these reactions happen? What provides the energy for a cell's endergonic reaction? Well, the answer is down here. It's usually the hydrolysis of ATP, or adenosine triphosphate. ATP is a relatively simple molecule, but within some of its bonds, it contains the potential for a quick burst of energy that can be harnessed to perform cellular work. So think of this molecule as the cell's primary energy currency, in much the same way that money is the currency that people exchange for things that they need. ATP powers the majority of energy requiring cellular reactions. As the name suggests, adenosine triphosphate is comprised of adenosine bound to 3 phosphate groups. So adenosine is a nucleoside consisting of the nitrogenous base adenine, so we see adenine out here, and a 5-carbon sugar ribose. So adenine right here plus ribose is going to give us adenosine. And we get the name triphosphate because that compound is attached to 3 phosphate groups. We have an alpha phosphate, we have a beta phosphate, and we have a gamma phosphate group. So phosphorus attached to 4 oxygen molecules. So together, these chemicals constitute an energy powerhouse. And the bonds that link the phosphate groups, so these bonds between the phosphate and the oxygen here, these are considered to be high energy bonds. And when these bonds are broken, their products have lower free energy than the reactants. So on the previous slide, we talked about the bonds between the phosphate groups being high energy bonds. So why is that important? What does that mean as far as what is going to be generated through this particular reaction when these elements are broken apart? So if ATP was to undergo a process known as ATP hydrolysis, we put ATP in the presence of water, what that's going to do is that's going to cause the breakdown of ATP, adenosine triphosphate, into adenosine diphosphate, so only 2 phosphate molecules attached to the adenine and the ribose backbone, plus a free phosphate group and some free energy. So we have to have that free energy because, again, remember, the first law of thermodynamics states that energy is neither created nor destroyed. So ATP is an unstable molecule. It's going to hydrolyze really quickly. In the presence of water, it hydrolyzes. Important to note down here, ATP hydrolysis is reversible. So we can take ADP and add phosphate to it using energy to make ATP. We're going to talk about how that's done later on in the talk. So we've talked briefly about the sodium-potassium pump, and now we're going to make it relevant in the context of ATP and ATP hydrolysis. So the energy derived from exergonic ATP hydrolysis is used by this integral protein pump. That's the energy that this pump uses to push 3 sodium ions out of the cell and bring 2 potassium ions into the cell. So let's talk about a potential quiz or a test question now, and let's make this applicable. So 1 ATP molecule's hydrolysis releases 7.3 kilocalories per mole of energy. Okay, so this is 1 ATP molecule's hydrolysis. If it takes 2.1 kilocalories per mole of energy to move 1 sodium ion across the membrane, so that's going to give us a delta G value of 2.1 kilocalories per mole, how many sodium ions could 1 ATP molecule's hydrolysis move? Give you a second to think about that. Okay, so again, apply the law of thermodynamics, the first law of thermodynamics. Energy can never be created nor destroyed. So we have 7.3 kilocalories per mole to work with here, so this number cannot exceed 7.3. So the most I can do is times 2.1 by 3, and that's going to give us 6.3. Right? So we can only move 3 sodium ions across the membrane with 1 ATP hydrolysis reaction. Again, the rest of the energy is going to be lost in the terms of what? And I hope you're saying heat energy. So again, energy is neither created nor destroyed. So with that reaction, we are going to get a 1.0 kilocalories per mole. Kilocal per mole loss of energy in the form of heat energy. A substance that helps a chemical reaction to occur is known as a catalyst, and the special molecules that catalyze biochemical reactions are known as enzymes. So enzymes can be defined as protein catalysts that speed up reactions by lowering the required activation energy. So now let me draw your attention to this picture over here. And in purple, you can see the reaction without the catalyst. So look at all this energy that's required without the catalyst. So you need a large amount of energy. But in the presence of the catalyst, we don't need nearly as much. Once we get to this energy level here, the reaction can be carried out. So there's a big difference in how reactions occur in the presence of a catalyst versus the absence of a catalyst here. So look at all this energy in here. Enzymes bind with reactant molecules, promoting bond breaking and bond forming processes. So we talked about the potential energy that's in those bonds and how that energy is released and becomes kinetic energy once those bonds are broken. And enzymes are very specific. They catalyze single reactions. Now let's talk about enzyme-substrate specificity. The chemical reactants to which an enzyme binds are known as the enzyme's substrates. It's also known as the reactants. So the 3D shape of the enzyme and the substrates determines this enzyme-substrate specificity. Substrate molecules interact at the enzyme's active site. So this is where all the action is happening. And enzymes can catalyze a variety of reactions. In some cases, two substrates bind together to form a larger molecule. In others, one molecule breaks down into smaller products. So now let's talk through this picture a little bit here. So both enzyme and substrate undergo dynamic conformational changes upon binding. The enzyme contorts the substrate into its transition state, thereby increasing the reaction's rate. Okay, so what does that all mean? So here we have a substrate and we have this large molecule. So there's two components. I'm just going to term these 1 and 2. This first reaction here is kind of like a lock and key. So on the active site of the enzyme, there has to be a proper fit for the key to come in. If there's not proper ridges for the key to fit, it's not going to bind. So this first step is the substrate entering the active site of the enzyme. Once the substrate binds to that active site, the enzyme is going to change shape. So there's a little bit of a conformational shape change. And that's going to start the process of some chemical reactions. Then the enzyme and products are going to form a complex, and you can see there is a color change here. So there are some type of chemical reactions occurring here. So we go from this green color to a blue and an off orange color. And then finally, the products are going to leave the active site once they have been modified. And you can see here we have two products that have been chemically modified and broken down from that original one larger substrate that we've had earlier. So how exactly do enzymes contribute to lowering activation energy? How do they help the chemical processes and chemical reactions actually speed up? So what they actually do is they help the substrate reach its transition state. Remember, the transition state is that unstable state. So these 4 elements here are very important for you to know. These are 4 elements of how the enzyme helps the substrate reach its transition state. So first, it can position 2 substrates so they align perfectly for the reaction. It can provide an optimal environment. So whether that environment needs to be acidic or basic or polar or nonpolar, it basically renders a perfect environment within the active site for the enzyme to bind to the substrate. It contorts and stresses the substrate so it's less stable and more likely to react. And finally, it temporarily reacts with the substrate. So it chemically changes it, it chemically modifies it, making the substrate less stable and more likely to react. After a catalyzed reaction, the product is released and the enzyme becomes available to catalyze another reaction. This is a really insightful video on the overview of enzyme function. So at this point, please pause the PowerPoint presentation and check out this link to this video. So it's very important to understand that enzymatic activity has to be governed and regulated and controlled, or our cells won't maintain the proper homeostatic balance that they need. So cellular needs and conditions vary from cell to cell, and they change within the individual cells over time. The required enzymes and energetic demands of stomach cells are very different from those of fat cells, and those are very different from skin cells, which are very different from blood cells, so on and so forth. And furthermore, a digestive cell works much harder to process and break down nutrients during the time that closely follows a meal compared with many hours after a meal. As these cellular demands and conditions vary, so do the amounts and functionality of different enzymes. And enzymes are regulated by 3 main factors. Okay, these are really important to know. Please make sure that you jot this down and you understand these factors, because you will see these on your quizzes and exams. So first, enzymes can be regulated by modifications to temperature and or pH. So if we put an enzyme in a warmer environment versus a colder environment, what do you think is going to happen to the speed of that reaction in most cases? Well, if you said the speed of the reaction would accelerate or speed up, you'd be correct. So remember, as temperature increases, so does the rate of molecule speed. And pH is really important. So some enzymes perform better in basic pHs, some perform better in acidic pHs, and some need a neutral pH. So modifications to the pH can change the enzymatic activity. Second, production of molecules that inhibit or promote enzyme function. So there are different molecules and compounds that facilitate enzyme function, known as agonists, and there are others that inhibit enzyme function, known as antagonists. And also the availability of coenzymes or cofactors, and we're going to talk about these. Some enzymes need the coenzymes or cofactors in order to properly carry out their enzymatic activity. As we talked about previously, enzymes can be regulated in ways that either promote or reduce their activity. And there are many different kinds of molecules that can inhibit or promote enzyme function, and various mechanisms that exist for doing so. Now let's talk about enzyme inhibition, and it's really important for you all to understand the differences between competitive inhibition and non-competitive inhibition, and the players, the competitive inhibitors and the non-competitive inhibitors. So let's begin by talking about competitive inhibition. So figure A here. Competitive inhibitors have a similar shape to the substrate, competing with the substrate for the active site. So I think of this in terms of a basketball game. Let's say there are two groups of fans. Let's say there's the fans of the Golden State Warriors and the fans of the Denver Nuggets, right? And let's say hypothetically the best team is Golden State Warriors, right? So the Golden State Warriors have the key to success, or the key to winning. So the Golden State Warriors are the substrate and winning is the enzyme down here, right? But only one basketball team can win the game. So if the Denver Nuggets win the game, that represented by this competitive inhibitor here, the Golden State Warriors can't win the game. So the substrate can't bind to the enzyme because it's being interfered with a competitive inhibitor. So again, the competitive inhibitor is going to bind to the same active site of the enzyme where the substrate should be. So another example is like a different key, a non-functional key that fits in the lock but doesn't turn the lock. So the key can fit in the lock, and that's going to prevent another key from going into that lock, but the key that's in there doesn't turn, so it doesn't facilitate any of the downstream reactions. On the other hand, we have non-competitive inhibition, so on the right side of this picture. In non-competitive inhibition, an inhibitor molecule binds to the enzyme in a location other than the active site. So again, look at the absence here. There's no binding at the active site, but what happens is the non-competitive inhibitor binds to a different site known as an allosteric site. And this binding is very powerful. It manages to prevent the substrate from binding to the active site. So some inhibitor molecules bind to enzymes in a location where their binding induces a conformational change that reduces the enzyme's activity, as it no longer effectively catalyzes the conversion of the substrate to the product. So non-competitive inhibitors, again, they're going to bind to the enzyme at a different location, and they cause a slower reaction. And we're going to elaborate on this concept a little bit more on the next slide. All right, so now we're going to layer on some more details about enzyme inhibition. So you've been introduced to competitive and non-competitive inhibition. Now I want you to understand this chart. You're going to see this chart potentially on your quizzes and exams. So first look at the purple curve, that's the normal enzyme, then look at the green curve, that is the substrate that's affected by competitive inhibition, and then in blue, the non-competitive inhibitor. So let's talk about competitive inhibitors first. So competitive inhibitors, they slow the reaction rates, but do not affect the maximal rate. So again, you can see here, the competitive inhibitor can achieve the same maximum rate as the normal enzyme can. Non-competitive inhibitors, they slow the rates and they reduce the maximal rate. The maximal rate is the speed of a reaction when the substrate is not limited. So again, you can see the slowing of the rate very early on, and you can see that the non-competitive inhibitor never achieves this maximum rate here. So how can I ask this as a test question? Well, I could label the normal enzyme B, A, I could label the competitive inhibitor B, and I could label the non-competitive inhibitor C. And I could say, which of the following is a competitive inhibitor? Which of the following is a non-competitive inhibitor? So please make sure that you understand this chart. And I mentioned briefly about the allosteric site in non-competitive inhibition. But please note that there's a lot that goes on at this allosteric site. The allosteric site can bind allosteric inhibitors, and it can also bind allosteric activators. So please know the difference between these two. Allosteric inhibitors modify the active site of the enzyme so that the substrate binding is reduced or prevented. So again, it's not binding to the active site, but it's binding to a different allosteric site, and that causes the active site to not have the capability to bind the substrate any longer. On the other hand, the allosteric activators, they modify the active site of the enzyme so that the affinity for the substrate increases. So allosteric activation is the opposite of non-competitive inhibition as far as what's going on at the allosteric site. So if an allosteric activator binds to that allosteric site, that's going to potentially facilitate the initial desired reaction to occur. Many enzymes don't work optimally or even at all unless they're bound to other specific non-protein helper molecules, either temporarily through ionic or hydrogen bonds or permanently through stronger covalent bonds. And two types of helper molecules are cofactors and coenzymes. It's important to know the difference between cofactors and coenzymes. So cofactors are inorganic ions, such as iron, magnesium, and zinc. So a cofactor for DNA polymerase is zinc. DNA polymerase requires zinc in order to carry out its reactions and interactions. Coenzymes, on the other hand, are organic molecules. So these include ATP, NADH, and all of the vitamins that you see in the picture here. And the coenzymes are predominantly derived from our diet. You've learned throughout this lesson that molecules can regulate enzymatic function in many different ways, and that there are many different players in maintaining homeostatic regulation. We've mentioned cofactors and coenzymes and organic molecules and inorganic molecules. And you may still be asking, well, how do all these metabolic reactions and these chemicals work together in the body to facilitate and maintain homeostasis? Well, the answer is it depends, and it's very complex. So there are many molecular pathways that are intertwined with one another, and they are very complex, and they have many activators and inhibitors. If you're interested in a very complex pathway, you can Google search the JAK-STAT pathway, J-A-K-S-T-A-T, and you can see all of these different molecules and players that facilitate regulation of this pathway. I want to talk briefly about feedback inhibition. Feedback inhibition involves using a reaction product to regulate its own further production. So the cell is going to respond to the abundance of specific products by slowing down production during anabolic or catabolic reactions. Such reaction products may inhibit the enzymes that catalyze their production through the mechanisms that you can see in the picture here. So metabolic pathways, they are a series of reactions catalyzed by multiple enzymes. And feedback inhibition occurs when the pathway's end product, which you can see in the picture here as isoleucine, inhibits an upstream enzyme indicated by the red bar here. So the end product isoleucine here is inhibiting the end product right there. So isoleucine will bind to threonine deaminase at the allosteric site and prevent threonine from binding to this enzyme's active site. And this effectively blocks the metabolic pathway. So when isoleucine levels decrease, threonine will then bind to threonine deaminase's active site, and the metabolic pathway will resume. This is an important regulatory mechanism in cells to inhibit the overproduction of a product. And this is just an example. I'm not going to test you on this specific example, but I want you to understand what feedback inhibition is in a general sense. So look up some other examples of feedback inhibition. Also look up negative inhibition, negative feedback inhibition. This can be seen through a lot of different cellular processes, and one that I studied extensively is the negative feedback regulation of the circadian clock. So that's something very interesting. So as we wake up, the same molecules and compounds that facilitate our waking are the same molecules and compounds that help us go to sleep. That's the negative feedback loop. So something that turns on a process also turns off the process. So I encourage you all to look at other feedback inhibition loops and look at positive and negative feedback inhibition.